Neuronal physiology. So these slides are going to build on what we learned in the muscle physiology slides. Many of the principles are similar, but we're going to go into more detail, and there are going to be differences that are unique to the nervous system. Hopefully, by the time you're done with both this unit and the last muscular physiology unit, you'll be able to explain how impulses are generated, transmitted to motor neurons or to muscle fibers, and then how that gets translated into an action by either the downstream neuron or the muscle fiber that it's stimulating. So on the whole, as one big organism, your body has a balance of positive ions and negative ions. It's roughly 50-50. Your body as a whole is electrically neutral. But that does not mean that you have the same number of positive ions on the outside of any given cell as on the inside of any given cell, or the same number of negative ions on the outside of any given cell and the inside of any given cell. You may have, for example, lots of positive ions kept on one side of a cell membrane and very few in the middle, or on the inside of the cell membrane. These ions, while even overall across your body, are not evenly distributed. And your body does that on purpose. There's an expression in biology that stasis is death. If you have everything, or equilibrium is death. If you have no movement, if you have a balance of things, that means nothing can happen. You need to have an imbalance of something in order to do some kind of work. You need to build up water on one side of a dam for water to be able to rush through and make electricity. Your body spends, or your individual cells will spend, a constant but small amount of energy in order to make sure that there is a dam built up across its membrane, that it has more of some ion and some charge on the outside of its membrane as opposed to the inside of its membrane. And then that way, if it opens up its dam and it lets those ions rush in or rush out, work can be done. You have a fast movement of something, and when you have movement of something, that can do work. The separation of ions has potential to do huge amounts of work when they rebalance, when they cross that dam, when they go through those suddenly opened channels. So maintaining this constant low-level energy expenditure, so spending this small but constant amount of energy, is worth it. Because when you open up the channels, you'll get so much more work done that you end up with a pretty good payoff for your investment. Now let's quickly learn a little bit of electrical terminology. This is the type of slide, by the way, that I see as a student and I start to panic. I'm not great with the physics stuff. And so when I see these terms, I think, oh no, this is going to be very physicsy. Don't be intimidated. The bare bones basic things that you need to remember are that volt refers to the potential of something to do work. The more you have the separation of ions, the more positives you have on one side of the membrane versus the inside, the more potential, the more opportunity there is for work to be done when the channels between them get opened. Current refers to the flow of those ions. So you have your membrane. Actually, you have your membrane, you have a channel, and you have ions. Lots of them on one side. We're just going to go with positive ions because I don't know, I like drawing pluses, I guess, and very few on the inside. How much more of these charges there are on one side of the membrane versus the other is referred to as the potential, and we measure that in volts. When you open the channel, and these pluses can come rushing in to equalize how many charges are on either side. How fast they move through that channel is the current. The resistance is pretty straightforward. We measure that in ohms. That's the hindrance to flow. So is there an obstruction? Are we talking about a membrane that doesn't transmit things well? How much something resists the movement of these things is the resistance. That's all we really have to remember for now. Volt refers to potential, and in more common terms like opportunity or ability. Current refers to how fast things are moving. Resistance refers to how much resistance, how much hindrance is there to that thing moving. There's 3 types of ion channels in the axolemma and on the plasma membrane of the soma and the dendrites that we need to know about. Chemically gated, more properly termed ligand gated channels, voltage gated channels, and mechanically gated channels. Chemically gated channels will open up because the right chemical bound to it. Think back to the muscles, right? You have a neuron ending, or you have an axon terminal that releases ACH, acetylcholine, into the synaptic cleft. That acetylcholine will bind with sodium-potassium channels, and when acetylcholine binds to sodium-potassium channels, those channels opened, right? Those sodium-potassium channels on the muscle fiber of the sarcolemma of the muscle fiber are ligand-gated channels. You're already familiar with those. Voltage-gated channels open and close in response to changes in membrane potential. So if you have a membrane here, and you have an imbalance of ions across the membrane, and your body is spending energy to try to build up more charge, or a certain amount of charge on one side as opposed to the other, a voltage-gated channel will open up when the difference between the inside and the outside is enough. So the more of one charge you have on the outside of the membrane versus the inside of the membrane, the more likely a voltage-gated channel will open up. Voltage-gated channels will open up at different points in time. So they need different amounts of voltage, of potential, of buildup of one ion or another in order to open. And we have mechanically gated channels, which are pretty much what they sound like too. These are going to open and close in response to deformation of something. When you take your fingertip and you push down on your desk in front of you, you have mechanoreceptors that as you squish the tip of your finger also get squished. That squishing represents a deformation of the shape of the cell. It used to be roundish and now it's flat. When you flattened out that sensory cell, it opens channels. You push on it, things open. You pull on it, things open. You pinch it, things open or close. So I mentioned that all cells will spend at least a little bit of energy maintaining a membrane potential, maintaining a gradient where you have more of one charge on the outside of the membrane or the inside of the membrane versus the other side of the membrane. Because when they open their channels and those ions can then rush through and equalize, that rushing through can do work. And it can do kind of more bang for your buck work than if it would have had to just generate its own. Your neurons spend energy, ATP, to export 3 sodium molecules for every 2 potassium molecules that it imports. This is an active transport mechanism. Sodium is positively charged. Potassium is also positively charged. By making sure that you're exporting more sodiums than potassiums, you always have an imbalance of more pluses on the outside, more positive charges on the outside as compared to the inside at a 3-to-2 ratio. So you have these channels where you have sodium, 3 sodiums that attach to a channel. And once those 3 sodiums have attached and ATP has released its energy, this is sort of cut off, but it will open up and release the sodiums out into the extracellular space. At the same time, when it's opened up like that, it can take potassiums and bind to the other side of the membrane, or the other side of the channel. When those sodiums have detached and those potassiums have attached, the shape of the channel will change. It'll drag those potassiums in with it, and it'll bring those two potassiums inside the cell. Again, this is ATP-driven process, so this is an active transport mechanism. Now, if you kept doing that forever and ever and ever, let's pretend those are the only channels that you had on the cell membrane. You keep doing that forever and ever and ever, you're just going to end up with a zillion positives on one side and less than positives on the inside, and then who cares? Also, if you had that going on and only voltage gated channels, you'd create a scenario in which those voltage gated channels would open up based on the concentration exclusively and not the cell's need to do work. So the neuron is going to spend energy to do this process, this active transport process, but it needs to make sure that it doesn't have too much of a buildup on one side. It has just the right number of pluses on the outside relative to the inside. So the cell membrane also has what are called leak channels. There are potassium leak channels that allow potassiums to leak into the cell very, very slowly, and sodium leak channels, which allows sodium to leak outside of the cell slowly. It does this along its gradient. I'm sorry, it's actually the other way around. Okay? So what you have is this active process of pushing molecules in and out, ions in and out, to keep an imbalance, but you don't want too steep of an imbalance. So you have these very few, very slow leak channels that make sure that your voltage, that the potential, the ratio of outside stuff to inside stuff, and the total volume of outside charge versus inside charge stays where the cell, where the neuron wants it to be. When we talk about charges going forward, we're often going to talk about the outside being positive and the inside being negative. In truth, the inside isn't actually negative. The inside of the cell isn't actually negative. It's just negative compared to the outside because we've been pumping positive ions out and keeping fewer of them on the inside. Relative to one another, the inside of the cell is kept negative compared to the outside of the cell. You also have chloride ions moving that also helps offset the movement of positive ions. These are negatively charged ions and also help to keep this balance steady. At this point, I would encourage you to pause this video and go into the other link on Blackboard that shows you the maintenance of resting membrane potential. Those processes I just talked about, active transport of sodium out and potassium in, coupled with chloride channels and leak channels, will help maintain the membrane potential at a specific place that the neuron wants it to be.